I would like to announce the open forum session of this morning. For those who do not know what the open forum is, so it's really the idea behind the open forum to give short insights into the diversity of interesting and ongoing projects. And as we did receive really many interesting submissions, thank you to all. So we have a lot of speakers, and so timing will be a little bit tight. So we have short 5 minutes presentations, and I will always introduce the speaker and already ask the following to get already ready. There will be no Q&A sessions, but you can keep your questions for coffee break, lunch, or the apéro later. All right, so then we start with Apex, with Elisaveta and Loreen. And I would like to ask Anna Karatala to get ready for the next presentation.
Hi everyone, we are here for you to present the organization for or Association for Early Career Polar Researchers. And I'm Lisa, and this is Lauren, and we are both PhD students at the EPFL. And our idea as the representatives is to get together the people is the first priority, and give the possibility for early career researchers to speak and to gain together and to build a community that will help them to exchange the ideas, also to learn maybe from more experienced colleagues. You can see that we are quite active, and we recently organized the event at Glacier 3000, so it was very spectacular to go there, and thanks to SPI, who is always helping us and supporting us, all our enthusiastic ideas, not only on words, but also financially, which is a very great thing, and we are very happy that we have such a nice people who support us. So here in the slide you have a picture, and this is probably the majority of our community. If you want to join to us, please follow the QR codes and join our WhatsApp group and our Slack group. So there would be also some events coming, the one that is in October, then the normal one that we usually have in November, going to Jungfraujoch, and we also plan to visit the collaboration visit to Alps Museum, but maybe Lauren will tell us more about the upcoming event.
Thank you. To advertise our currently ongoing submission for fieldwork pictures, we're hoping for many more people to submit. So if you are already a member of Apex, you probably received this. And we are hoping that you will also submit your fieldwork pictures. It's really the broad spectrum of active people in the field, but also some nice snapshots of the beautiful scenery that we have in these environments. If you are not part of Apex, then please join us and submit your pictures. So you can submit up to 3 pictures, add a short description, make sure that you are allowed to share the people or publish it with the people on it. And then we have in October our photo contest exhibition with prizes, also sponsored by SPI, and we're hoping to see many of you. So please sign up, spread the words among the early career scientists in your groups, and we're hoping that we will earn some new members today. Thank you.
Thank you very much, Lisa and Loreen. So then I would like to give the floor to Anna Karatala from EPFL, and I would like to ask Alina Begley to get ready.
Thank you. Hi, everyone. Thanks for allowing me to present a project I did last year, supported with an exploratory grant of the SPI. The aim of this project was to understand a little bit better the environmental adaptation of bacteria communities in Arctic lakes in face of environmental changes that we know these environments are experiencing. And the project relied on one main idea, which was, how about we zoom out from lake water columns and only study the water columns, and we zoom out and try to understand also what other interplay and connections might be happening. So if we really look and we zoom out to lakes, we have the air and air bacteria that are interacting with the water column, but also there is a temporality of bacteria that are being deposited on sediments in a way that environmental change might be faster in the air and slower in the sediment, but also those bacteria that we find deeper in this complete column might be older than those that we find at the top layers. So looking at this new lake column, we might get further insights on the environmental adaptation of bacteria communities in these environments. So to actually explore this idea in more detail, I did some field work in 2024 in a region that I knew had higher diversity of bacteria communities from previous work that was supported by Christiane Leister in 2023. So I went to this lake and I invested myself in sampling water, but also aerosols and sediment cores from this lake in 2 different times of the year, to get further insights on the spatiotemporal variability. And I'm still analyzing the data, of course, but I can share some preliminary ideas that I learned from this project. And the first is that each compartment of this real lake column might represent a very different ecological niche. So, for example, ice cover in the lake in June showed lower concentrations of certain elements and higher concentration of nitrogen-related elements, for example, ammonia and nitrates, suggesting that ice cover, for example, might have more important ecological role than initially thought. Then these differences in the environmental variations of these compartments do actually translate into differences in the bacteria communities, for example, in terms of their abundance. So I was able to identify different biomass of bacteria in each different compartment, so lower in aerosols and higher in the ice cover precisely. But also spatiotemporal variability, suggesting that the response of bacteria communities to environmental changes is rather timely. And then finally, this is still under development, so the results I'm showing here are from 2023, but similar analyses are being done currently with the new data. We know that the bacterial communities that are hosted in each different compartment are actually different. There is different bacteria that are associated to different areas of this column. And not only that, but also different metabolic capacities are also detected in each compartment. So this overall shows or enforces the idea that studying all these different compartments might confer further resolution and understanding environmental adaptation of bacteria. So if you want to know more, we can discuss later at the coffee break.
Thank you very much, Anna. So, Alina, the floor is yours, and I'd like to ask Christian Haug to get ready.
All right, at EMPA Dubendorf, we monitor trace halogenated gases in air. Halogenated gases contain chlorine, bromine, fluorine, and/or iodine, and are almost entirely man-made. They're used for air conditioners, refrigerators, and such. And the issue with a lot of these is that they have either very long lifetimes in the atmosphere, they can destroy the ozone layer, or they are very powerful greenhouse gases. That's why many of them are regulated under the famous Montreal Protocol. Air is mostly nitrogen, some oxygen, and a small amount is CO2, right? 400 parts per million. Well, halogenated gases are trace levels, so they're 1 million times lower in concentration than CO2 at parts per trillion. That's why we have a special instrument that you see on the top left that we use to pre-concentrate the air before we do the measurements. And we have basically two major ways of measuring air, either in situ, that means we take the air directly at our instrument every hour, for example, at the Jungfrau Joch High Altitude Research Station, or we collect the air in flasks, like you see next to the penguin, and then measure them later. So we have a list of about 60 substances that we continuously monitor together with the A-gauge measurement network. And an example you see here is volatile anesthetics. So on the top, we have desflurane, which is used in human medicine, so if you go to surgery. And on the bottom, we have isoflurane, which is primarily, but not only, used in veterinary medicine, so if your dog goes to surgery. And what you see on the top of the graph are our in situ data from the Northern Hemisphere. And on the bottom in green are the flask measurements. So we've been getting, since about 2010, through a collaboration with the King Sejong Research Institute or Research Station in the Antarctic, we've been getting weekly flask measurements since about 2010. So that's how we end up with these long data sets where we can see the concentration in the Northern Hemisphere and the Antarctic. So a couple really cool things that we can see are seasonal trends. So you see that the concentration is lower in the summer because there's sunlight, which effectively depletes the compounds, and it's higher in the winter. And then we also see long-term trends. So desflurane and isoflurane were increasing in the atmosphere until about 2020, and then desflurane started going down because many hospitals stopped using desflurane because it has a long lifetime in the atmosphere and is a powerful greenhouse gas. It's going to be regulated by the EU starting next year. And then we also see the difference between the northern and southern hemispheres. So as most of the pollution is happening in the northern hemisphere, it takes maybe 1 to 3 years before that pollution is circulated all around the world. So that's what you see there. So usually we have to monitor substances that we know ahead of time. But since about 5 years, we have a great instrument from the Swiss company TÜV. It's a TÜV instrument that allows us to basically measure everything in the air, and then we can go back in time, and based on how the compound fragments, we can reconstruct them and figure out what the compound was. So together with the Swiss Polar Institute, we were able to collect 1 year of flask measurements in the Arctic at Ny-Ålesund to complement our Antarctic flasks on the bottom right. And actually what we found was some new short-lived chlorinated species that are used in the production of polyvinyl chloride, or PVC plastic. We detected them in both polar regions and have seen seasonal trends for these compounds. So the next step that we want to do is quantify emission sources for these and see what is their role in ozone depletion, what role might these compounds play? And then finally, continue to use this instrument to basically monitor the polar regions for new anthropogenic compounds. Thank you.
Thank you very much. So I'd like to pass the floor on to Christian and ask Heather Corden to get ready.
Thank you. Yeah, so hello everybody. My name is Christian Haug from the University of Fribourg, and I'm here on behalf of my colleagues Colin Mollaret, Cécile Pellet mainly, who managed to acquire this new cost initiative, a cost action, which will start in October. So it's very new. And the topic you can read is the coordinated and standardized monitoring of permafrost response to climate change. So now you can argue, isn't that existing already, a network of permafrost observations all over the world? And so far, unfortunately, that's only being done with borehole temperatures, so there's a borehole temperature network. But for example, in Switzerland, we have since already 20 years, 25 years, we have the Swiss Permafrost Network, which combines also other monitoring elements, for example, velocity of movements, as we have seen in the talk by Thomas Thurbuch before, and also geophysical measurements like electrical resistivity. There was also a project funded by the SPI in the last years. So there are many, many more variables which we could measure, be it from space or be it on the ground, and these data need to be somehow, and now we come to these working groups, which you can see here, they need to be identified. I mean, you could argue borehole temperatures is clear, but is it important to monitor the water content in permafrost areas? Is it important to monitor the movement for every slope? Is it important to monitor, let's say, the mineral content or the density, etc.? And for the people who are working with the monitoring methods, be it from space, be it on the ground, these variables are important, of course, because it defines which geophysical technique you would like to install in the ground, etc. So that's what would be the first working group, and the second is then data acquisition standards, homogenization standards, processing standards, and so on. And of course, the final aim would be, and again, one could argue, why do we need this? But it's not existing so far. Unfortunately, we would like to have a permafrost state assessment over all of Europe. This is because it's a European tool. It's a funding tool. These cost actions, but as you can see by these blue squares here, it's not only being, addressed in Europe itself, but let's say by European researchers in combination or in collaboration with international colleagues. So we can include very easily the whole of the Arctic and the Antarctic, which is indicated here, although the main networking activities will be by the European institutions. But this is what COST is all about. It's open to everybody, so technically everybody in the room can apply with a click and participate in this COST action, and then being invited to networking activities. So the main aim or the main focus, as you can see on the right-hand side, COST provides funding for research, coordination, training, and networking. So it's really intended also for the young generation, for early career scientists, enable them to take part in all the, let's say, networking activities the more senior researchers can fund through other sources. It's not intended to fund scientific projects by the scientific researchers. It's also not a lot of funding involved. But it facilitates exactly those kind of collaborations and networks and generating collaborated outputs. So this is what we would like to do in the next 4 years. It's also quite a bottom-up procedure. So what we did in the University of Fribourg with a lot of international colleagues, so these are on the left-hand side, this network of proposers from 24 countries all over the world. We proposed this COST action, and in October will be the first meeting, and then the grant holder institution is elected. So we volunteered, but in Fribourg, but it's still open. So if somebody else wants to take over the leading role and all the work, then you're very welcome. So it's really the same goes for the working group. So if somebody volunteers to be a work group leader, we are very, very happy if these persons will contact us. So the email address is permacost@ödfr for the moment, but you will find this COST action also on the COST web pages of the European Union. So it's all there, and you can directly apply there to be part of the network. And we hope also to inform you regularly in Switzerland, of course, but also elsewhere about the ongoing of this COST action. Then you can participate also later. So I think it's a very unique opportunity for the permafrost community. It's a very unique opportunity for Switzerland because we have already this quite nice working and funded permafrost network, but which is very, very rare all over the world. So there is one in Canada and one in Norway, and that's basically it. So this is what we would like to do. We would like to expand these networks all together to have a real worldwide and, in this case, European network of permafrost. Thank you.
Thank you very much, Christian. Then I would like to ask Benjamin Hoffmann to get ready, and Heather, the floor is yours.
Thanks.
I'm here from the group of Alexi Byrne at EPFL, and I'll be presenting the OACA project. So OACA is an ERC synergy project with collaborators at EPFL and also in France. And I first introduced the project a bit, so the graphic at the top. The acronym stands for Atmospherics Water Cycle over Antarctica. I would like to understand this in the past, present, and future. So in the present, we have a rather large observational transect between Dumont d'Urville on the Antarctic coast and Concordia on a high plateau. And along this transect, we'll make meteorological and also water isotope observations, which will then be used alongside models and improved parameterizations to look at the past and the future. So in the past, we want to revisit ice core interpretations using water isotopes, and in the future, we'll produce an ensemble of model forecasts for the future state of the ice sheet. So a current update of the project, the instruments were deployed in the most recent summer season, 24 to 25. The transect consists of 4 sites with these box container stations, which you see on the left. 3 of them are remote, so completely energy autonomous in theory. And the 4th one is situated at Concordia, so plugged into the station power supply. And at each of these 4 sites, you have 3 weather radars at different frequencies, a LiDAR, and also isotope instrumentation, which can measure water isotopes both in the water vapor in the air, but also in hydrometeors, snowflakes. These container stations were deployed in a tractor and sledge traverse with the French Polar Institute, and they'll be in place for 3 years. In addition to these container stations, we have more instruments at Dumont d'Urville, which is the station on the coast. And these are instruments with higher power or operational requirements, such as a big scanning weather radar and some additional weather radars at different frequencies. We also have a snowflake camera. And with these, we can link the things we see with the radars to the processes we see on the snowflake pictures. And just a quick taste of what I've been working on. So within this big OACA project, I'm mainly focused on the radar observations in the present. And I've been looking at a case study in February 2025. And you see in the upper plot, visualization of the total column water vapor anomaly compared to a February mean. And you see a big, warm, red, moist intrusion traveling inland from the coast, also going over all our sites, which is rather nice, sort of proof of concept of the project. And you see in the radar plot at the bottom, we can see this event, this warm intrusion bringing lots of moisture and precipitation, and we can observe it at different points along the transect. And I've been looking into the radar variables. We can learn lots of information about microphysics and as this event travels inland, the different processes leading to snowfall. Finally, a quick update for the project as a whole. So the instruments have been in place for 1 year. Unfortunately, not all of the remote energy systems fully survived the year. Some of them didn't work through the winter. So this year there'll be a maintenance campaign with the main aim of rebooting and improving these energy systems with the hope that in the future years we'll get complete years of observations in these remote sites. Thanks for listening.
Thank you very much, Heather. So I would like to ask Virginie Marquez to get ready and give the floor to Benjamin.
Thank you. So my name is Benjamin Hofmann. I'm a group leader at Transdisciplinary Lab of ETH Zurich since last month. And maybe we can switch the slide. Is that here? Yep. many of the things we hear today, they are connected to changes in the environment, in the Arctic, Antarctic, in the Alps. And I want to bring in the social element into this discussion, because many of the landscapes that seem empty at first look, they are either inhabited by humans or they are in one way or another connected to human activities. So this is why we thought these regions where climate change is happening so fast and all the environmental changes that come with them, maybe they are nice laboratories to see whether also the relations between science and society are changing. Are researchers starting to position themselves differently in relation to societal actors or not? That's the question that I'm exploring together with Ariane Wenger, who is a postdoc in this project that is funded by SNSF. It's called AnthroRelate. And we do two things in this project. The first thing is we want to rethink science-society relations. So far, they have often been framed as dichotomies. Is the science problem-oriented or is it solution-focused? Is it distant from society or is it engaged with stakeholders? And we want to go beyond that. We want to focus on two dimensions. The first one is long-term mobilization of stakeholders. Are researchers able to develop long-lasting relations with stakeholders, building long-lasting networks well beyond the project lifetimes? And the second dimension is critical engagement with system structures. What we mean here is, do researchers stop with making some policy recommendations, or do they go further? Are they able to raise critical questions about what are the decision-making structures behind that? For instance, are our decision-making structures fit for the purpose of climate adaptation, or do we need to change something more fundamentally? The second thing that we are doing in this project, we want to sort of understand what makes science-society relations transformative. And we say that if you score high on these two dimensions, right, you would have potentially transformative relations with your stakeholders. But what are the influence factors behind it? And we interview researchers and stakeholders in two regions. One is Arctic Finland, so the region of Lapland, and the other region is the canton of Grisons in Switzerland. And I can share some first insights from interviews in Finland. And what we saw is that what seems to drive transformative relations is local embeddedness of researchers. So we saw researchers who do projects on climate adaptation in reindeer herding, for instance. And several of these researchers have a background themselves in reindeer herding. So they either worked on these farms or they live in communities that practice that. So they bring the concerns of these communities into their research, which makes it quite transformative. Then we also saw that European funding is a push, because in Horizon and in Interreg you have all these requirements on deliverables that are oriented towards society, so that pushes researchers to engage in that space. Then we also saw that involvement of social sciences is often critical for this dimension of critical thinking, asking about system structures and so on, not only because social scientists organize you a nice workshop with stakeholders, but they're also able to bring that sort of critical mindset and political mindset also to the project. And then lastly, indigenous community played a role. So you have the Sami minority in the north of Finland, and that raises a lot of questions about involvement. So for instance, they have a reindeer herding association on the national level, but the Sami people, they went out of that association. Now, as a researcher who does research on reindeer herding, whom are you going to collaborate with? There were some who said, well, we follow the law, it's the National Reindeer Herder Association. There were others who said, we try to balance, we integrate both of them. And then there were others who say, we want to bring the voices that have been sidelined to the table, to the fore, so we only work with the Sami. And that's quite interesting tensions, I think, that you can observe there. If you're interested to learn more about our project, scan the barcode, and we will also organize a workshop early next year to feedback our results to researchers who are looking into how can I make my research more transformative. So please talk to me if you're interested. Thank you.
Thank you very much, Benjamin. So then I would like to ask James Ellison to get slowly ready and give the floor to Virginie.
Hi, everyone, and thank you. So I'm Virginie Marcus, and I'm working at the VSL and at ETH. And I'm mostly working with environmental DNA and studying fish. But for this project, we have a collaboration with the environmental physics groups at ETH, and particularly with Maaike Vogt. And there we took the opportunity of the research vessel Foral, who's very recently roaming the northern hemisphere and the high latitude areas to try to understand what are the patterns that we see with the plankton biodiversity, and how then can we link it to the higher trophic levels, the fish that people rely on for their livelihood. So there, it's very recent, so I have no results to show you, but I can show you what we did. So this is from last June to July. This was the path of the research vessel Foral that left from Canada, that went around the coast, and then they crossed the sea all the way to Ilulissat in Greenland. So this is the path that were there, and the method that we used to see what type of plankton we had there, and then fish were the environmental DNA, where we can get basically anything we aim for. So we'll there target the planktonic communities and the fish, and a new method that is emerging. So was aimed as the citizen science method to try to ease the collection of data that we can get, so that it's easier, faster. And that's the planktoscope. And the planktoscope works that we can use a plankton net, we deploy it easily from a ship, and then we have this tiny box there, which is in itself a very tiny instrument that has a small computer that is basically taking pictures, and there we can take images of the plankton of a certain size, and then we upload all the data online, and then AI is pre-training everything that we can identify. So from these two methods, we get a broad overview of biodiversity, we get images from the planktoscope, and we can get abundance as well using the plankton net. And the goal here of the project is to put it all together, and then to link what we have in terms of plankton, in terms of fish, and in terms of the environmental conditions. So, for example, one pattern that we have in high latitude is that we see that we have a larger size of organisms when we get up to the northern hemisphere and the colder temperatures, and we have smaller size of organisms when we are warmer seas. And there, with the gradient that we have in this project, we will be able to see how the communities or structures, weight changes, and what type of gradients we have, both from the coast to the open sea, and then in terms of temperature, because it covers a very large gradient. And then we hope to connect all this planktonic communities towards the fish communities. And so with that, I think the ETH Versailles and Swiss Polar Institute for the funding, and of course the crew of the research vessel Foral. And stay tuned for results.
Thank you very much, Virginie. Then I would like to ask Sandra Wells to get ready and give the floor, it's already there, to James Ellison. Voilà.
Thank you very much for having me today and for braving the terrible weather that we all faced. So, Antarctica, the best place in the world to do direct air capture. Let me try and convince you of this statement. I don't think this audience needs convincing that reducing carbon dioxide levels in the atmosphere is a positive thing, and direct air capture is a very tantalizing way of doing it. It can remove historic emissions, it can mean we can all still continue to fly, but at the moment it's extremely expensive, principally because it consumes an outrageous amount of energy to do the capturing. Now the energy... The energy required to do this capture is a relatively simple equation to calculate. It's proportional to temperature, which is an enormous win for Antarctica. It's 25% colder than almost anywhere else on Earth. But also at these low temperatures, the CO2 liquefies rather than becoming a supercritical fluid. So you also win on the amount of pressure that you need to apply. And so you halve the theoretical amount of energy that you would need to capture the CO2. The Antarctica is also one of the richest places in the world for wind energy. It dwarfs almost anywhere else. I should acknowledge this is from the ERA5 data set, the weather data. If we then do some modelling to calculate how much CO2 we could capture per kilometre square of area, we see that Antarctica dwarfs anywhere else in the world. I had to rescale this axis so that the northern hemisphere was not just entirely blue. Now, you can do things to improve this. This is for one system. If you include things like a dehumidifier, the Northern Hemisphere becomes a lot more promising again, but that adds additional complexity and cost to the system. Speaking of humidity, Antarctica is extremely dry. This is an enormous win for the materials we could use for direct air capture. So this is a zeolite. It's the same stuff that might go in your cat litter tray. It does an amazing job of capturing carbon dioxide at low temperature, but it also captures water very well. When there's no humidity in Antarctica, this is no problem. We can use the zeolite, which is very cheap, very commercially available. There are some problems potentially with capturing CO2 in Antarctica. The first being that it is extremely desolate and thousands of kilometers from any sort of industrial base to build your carbon capture module. Now, the modules are quite dense. They capture about 10,000 times their own mass over the course of your lifetime, so you could move them in from somewhere else. But this is still an open question whether that's even possible. It's also not particularly obvious where you would store the CO2 once you have captured it. Now, from ice cores, we know that you can store CO2 in the ice, but it's not obvious that you could do that for long durations or in a man-made way. And finally, a big question is if any of this is actually legal, and as a community and as a society, whether we'd actually be able to tolerate doing this. I do not know the answer to this. I'd be very interested to hear your thoughts on whether this is permissible both by Antarctic treaties and if it's something we would want to do. I will leave you with the thought, though, that for every 1 square metre of land that we use for CO2 capture, 100 metres squared of ice doesn't melt. So it is very much a net positive for doing direct air capture. What are we doing to test any of this? So this winter, going to Jungfrau Joch with a prototype to see if the modules can withstand the harsh environments akin to Antarctica, and also whether we can realise any of these at the moment theoretical energy gains in such an environment. Thank you very much for your attention. I look forward to meeting you over the conference.
Thank you very much.